Bioinformatics.
Today I want to talk about genome assembly. Now, one of the downsides of this remote learning environment is we didn't get to do one of my favorite labs this semester, which has nothing to do with coding. It's actually this idea that I have adapted from a great bioinformatician in Titus Brown from UC Davis, where we take a popular paragraph from American literature that is full of typical issues you find in genome assembly that we'll get to, repetitive elements, and pretend to disassemble it into the equivalent of next-gen sequence data. In other words, random fragments of some number of depth. And so here is the paragraph that I disassemble typically and give to you guys in the form of reads. And so typically it's 20x coverage, and the reads vary in length. They can also be paired-end or short-end, and they also have low levels of error. And so the paragraph is from A Tale of Two Cities. You can read it yourself, but most of you may have heard at least parts of it. You know, it was the best of times, it was the worst of times, it was the age of wisdom, etc. And so in class, you guys work together in groups and cut these things out and use tape and scissors and try and reconstruct this paragraph, which just from the reads, it's not always super clear where it came from, and try to get this paragraph reassembled using reads that are 10 base pairs long or 11 base pairs long, and think about different genome assembly methods. And so I bring this up not to make you feel like you missed out, but because I think some of the examples using words rather than nucleotides make the most sense. So I want to give you some context for the examples I use a little later on in this lecture. So why is assembling a genome important? So there are so many genomic methods that you really can't even do unless you have some kind of reference, often a genomic reference or a transcriptomic reference. And it's just a barrier to entry that you need for whether it's looking at SNP/SNVs, whether it's looking at gene expression. There are so many things that you need to do. Some people will just go out and want to sequence genomes just because. No one's ever sequenced a fern genome, if that's something that motivates you. And so I kind of liken this to some of the motivation behind some of the great mountaineers. George Mallory, for example, when asked why he wanted to climb Mount Everest, said because it was there. And so it's not always the greatest reason to sequence a genome, but it's something that actually happens out there in the world. That said, genome assembly requires a lot of sequencing, which is very expensive, although it's getting cheaper. So it's best to have some kind of underlying research question to motivate you to assemble a genome because there is kind of this monetary investment. So examples of what you can do once you've assembled a genome, again, SNP/SNV discovery, these are things that you've done in lab. If you're interested in transcription, you could map RNA sequence reads onto a genomic reference or genomic, let me start over. If you're interested in gene expression and intron-exon boundaries, if you mapped RNA sequencing data to a genome assembly, where there are gaps between the reads that map and reads that don't map, you could infer those to be intronic regions within genes. Challenges, though. Genomes can be big, particularly eukaryotic genomes. You can end up with genomes that are anywhere from 1 megabase, in other words, 1 million base pairs, to 50 billion base pairs, or 50 gigabases. These are all within the realm of possibility if you're looking at eukaryotic genomes. To put this in context, if we're thinking about just a 1 gigabase genome, a very average eukaryotic genome, in book form, like if you were to try and find it at the library, it would take 1,000 books that have 2,500 characters per page and 400 pages to contain one copy of the genome. Keep in mind, when we sequence, we typically want somewhere in the ballpark of 50x coverage, 50 times the amount of base pairs sequenced relative to how many are in the genome. So you could really think of that data set for assembling that genome, that 1,000 books back together, would have to be assembled from tiny cut-up pieces of 100 to 250 base pairs after shredding 50,000 of those books. And so that's a really difficult challenge. If you did the paper assembly in class, if we were doing this in person, you'd see that even doing one paragraph is challenging. So It's a very difficult problem. It's made more difficult by the complexity of genomes. Again, particularly eukaryotic genomes are full of repeats. If you were doing an assembly of all the songs produced in the last 2 decades, you would just by chance run into phrases that repeat throughout different songs, even though they aren't within the same song. If we tried to piece these things together and we were some alien race, we wouldn't know that certain songs don't go together. And you might end up assembling na 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 across multiple songs and stringing them all together. Looking at it from a genomic perspective, repeat regions, here we can see in red and yellow. If we imagine these as being identical sequences and we were to assemble these naive to the fact that they are repeats in different areas, we would actually assemble something that looks like this. And the other middle piece here would end up being assembled separately, and we would miss out on some of this actual true biological complexity. The way we get around these repeat regions varies, but the underlying challenge, particularly when they're repeats that are tandem, meaning really long stretches of short repeats, you can think about microsatellites, things like that as well, if they go super long, is that our reads are only going to be able to go all the way across them, bridge them, if you will, if they're long enough to span it while still maintaining some level of unique content. In other words, if we have some upstream unique content, some unknown length of a repeat, and then some downstream unique content, we can basically bridge our way across with our reads until we reach the point where there's no more unique information on one end. If we end up with reads that only contain the tandem repeat, do they go here? Do they go here? Do they go here? It's impossible to know. So when our repeats extend past the length of our read length, we start to run into issues. Some of this can be overcome if they're imperfect repeats, where you get some polymorphism throughout, so there is some unique information for placing your reads. However, it can be really confounding if you've got low coverage, in other words, a low number of mapping reads to that region, and you're also trying to account for sequencing error, because is this polymorphism here true in your read, or is it just an artifact of sequencing and it's a miscalled base? So this also is not a perfect solution. Other difficult issues that we run into are non-uniform coverage. So I talk about we typically want 50x coverage. In other words, on average, for any base pair in the genome, you would like to have at least 50 reads stacked up above it. That is absolutely not how it works in real life. There is random chance. There are differences in biases associated with library prep and GC content that can, rather than giving you a really consistent amount of reads mapping to a region across the genome, you can end up with these kind of peaks and valleys where you have regions of really low coverage, where you have little to no information for helping you assemble that area. So how do we actually assemble the genome? I'm going to talk about 2 methods very briefly that aren't really used anymore, and then talk about kind of the current best practices. So a greedy algorithm, we've talked about these before, is an approach where you just make the best decision for every single decision without thinking about the big picture. And so in the case of genome assembly, a greedy approach would be to take all your reads and then just pairwise align the 2 with the most similarity, and then take the next most similar, and then the next most similar, and the next most similar. And so you're just constantly taking just the very best potential alignment for every single step. Now this will work for getting some decent sized chunks of assembly, but you run into this issue of local optima. And so this is prevalent with all greedy algorithms, where you're always going to start at some random point relative to where you actually want to go. And so it could be here, it could be here, it could be here. And every time you make a decision between which alignment to do, you're going to make the one that increases the size of your assembly or the quality of whatever thing you're trying to do if it's another type of greedy algorithm. And at some point, you're going to reach a point where there's no decisions to make that will improve the quality of what you're trying to accomplish. That does not mean that it's done. It just means that you have reached what we call a local optima or a local peak, where there are no more better decisions, but you're not at the best decision. But you're incapable of getting there because you can't see the big picture and the ability to traverse these valleys for decision-making purposes. And so that's kind of a problem with all greedy algorithms and why they're not really used for genome assembly very much anymore. This is the method overlap or layout, consensus. This is the method that pretty much everybody uses in class when we do the paper assembly of that paragraph, where you take all your reads and you lay them out and you try and figure out how they overlap and paste them all together and tape them onto the table and fun stuff. It's very similar to a multiple sequence alignment. And it works pretty well. It's not a bad method. The problem is it's really computationally expensive, and at a certain point, the data sets we're working with to assemble large genomes just outpace our ability to perform this type of method. So that brings us to how most assemblers work now. They use what's called a De Bruijn graph. This is kind of a computer science abstract way of thinking that dates back to the 1700s. You wouldn't think you would learn a little history in bioinformatics, but you will. So back in 1700s, there was this notable problem in mathematics that a lot of philosophers like to debate of these 4 land masses that were connected by the 7 bridges in Königsberg. And the idea was that people would debate how you could mathematically prove that it was impossible to navigate the entire city crossing each bridge only once. And so you can sit here for an hour and try and figure out how you could cross all these bridges. It's an impossible task. But it was not formalized as far as a analytical conclusion. It was just trial and error. And so mathematicians wanted to find a way that they could prove this. And so the person that found this, in other words, found a solution to proving that there was no solution, was Euler. And so what Euler did is he came up with this idea of what would eventually become graph theory, basically. And so he converted this kind of idea of these 4 land masses and 7 bridges into 4 nodes connected by 7 connection points. And now that he had this kind of graph way of thinking about this, he determined that the only way to actually make this walk, and it would later become known as a Euler walk, to cross every bridge only once, is if every node was connected by an even number of connection points or bridges, except for both the beginning and the end could be odd numbers. And so in the case of the 7 bridges of Königsberg, they're actually all connected by odd. And so it does not fit the graph criteria of having all even except for the terminal points, the beginning and the end. Here's an example of one that actually is capable of a Euler walk, because if you look, there are only 2 nodes that have an odd number of connection points. So this is really what he ended up formalizing and becomes the basis for De Bruijn graph assembly. So how do we get to that from a bunch of sequence reads? Well, first thing we do is, just like in BLAST, we want to make a bunch of k-mers. K-mers will be a little bit larger, typically 12, 20, 30, 40. We'll talk more about that. But we'll basically use that sliding window of k to disassemble our reads into shorter, highly overlapping k-mers. Now, the reason we do this is because finding overlap between k-mers is much less computationally intensive than the whole reads in and of themselves. The downside is the value of k, the size of your k-mer, can have enormous consequences for your assembly. And so it requires quite a bit of trial and error to figure out what the best k is for your organism. There is no universal answer. Depending on the size of your genome, smaller k's might be better. Larger genomes might need a larger k. The overall goal is that k is large enough that it's not going to share overlap with k-mers that it's not actually next to in the genome. In other words, they're unique in their placement, but not short enough or not so long that they don't do that. And so there's this kind of fine balance between being specific but not too specific enough, as well as computational intensity. So how do we translate k-mers into a De Bruijn graph? So let's say we have these 2 sequences, this AACCGG and then CCGGTT. We're going to disassemble these into formers, so k equals 4. So you can think about what that would look like. It'd be AACC, and then it'd be ACCG, and then CCGG, and then the same thing down here. What we do is we line these things up and overlap them as best we can. So here in this box C, we can see here's the first K-mer overlapped all but one base with the second K-mer overlapped all but one base with the third K-mer, and so forth. Now, if we were to draw this into a graph like Euler did, it would look something like this, where every node is a K-mer, and the bridge connecting it is basically the addition or change of one kind of shift in that window or base pair. So here it's AACC, and then here we have this shift where we lop off the front A and shift over and add a G, and then we add another G, and then another T, and then another T here. And so that would be reflect this kind of actual sequence here. This actually does kind of look like what the overlap consensus approach that students use doing the paper assembly. And we'll see where this kind of deviates as you start to how you use these kind of overlaps in a few minutes. So if we want to think about either nucleotides or the paragraph from the paper assembly, we can think about situations where you actually have more complication, where you've got two sequences that are highly similar but diverge at one piece. So here, highlighted in red, there's a difference between this sequence on the bottom and the sequence on the top. This is very similar to how we have a lot of similarity in it was of times, it was the, and then it could either go, it was the worst or it was the age. And so we can graph this out using branches. And so here we've got our K-mers. This time it's K equals 14. I'm only going to show the bases that are added just to keep this thing short. So it's this first big chunk, and then there's a C that's shared, a T, T, G, A, C. And then once we get to this C point, you can either have overlap with K-mers from this or K-mers from this other read. And so rather than try and figure out which one's right, they both could be right. And so what we do is we just branch it. And so one path goes A, G, G, C, G, A, and one branch goes G, C, G, G, A, T. Okay? And so this provides us a lot of information. Now, sometimes this is biological, and sometimes it is just noise. And so for simplicity's sake, I'll show you when it's just noise first. You could imagine if you had a bunch of reads, K-mers from those reads, and one of those reads had a sequencing error that was one or two base pairs long. You could imagine it being all the same as it's doing its overlap, and then just one or two base pairs that diverged that were different, and then stopping that divergence. This is what we would call a spur. This is something that's very easy to identify from our graphs after we assemble all of our K-mers and go, that's probably not biological. It's only one or two base pairs difference. It's not supported by a lot of reads. It's probably just sequence error. So we can kind of many times prune off these spurs as well and move on to thinking about biological issues. Biological issues that might be reflected in the graph are if we have a divergence, so basically sequence divergence between some of our reads, that then eventually reconverge in their overlap. We call this a bubble. To think about a biological example, if you were sequencing through a gene and assembling it, and that gene had two possible alleles, you would run into a point where one allele had a different sequence for a certain stretch than the other. And so the upstream regulatory stuff would all be conserved. You'd hit a region where both alleles had different sequences, but then eventually you get back to a point where the rest of the sequence is the same and it converges again. And so this bubble is reflective of polymorphism between the two alleles, for example. We talked about repeats earlier. Those are one of the biggest challenges and are often represented in this graph as a frayed rope. You could have two regions that are very disparate within the genome. They're not anywhere close, and they could be having their overlap line drawn, and at a certain point they both hit a transposable element that they both share. And so they're going to get drawn together because there's a lot of overlap between that repeat sequence. Even though in the genome, these two, this start point and this start point, are nowhere near each other, this repetitive element will be pulled together during assembly. And that's something we have to try and fix too. So These can start to kind of look kind of funny. So here is a De Bruijn graph of part of that paragraph. Here you can see it was the best of times, it was the worst of times, it was the, and then you get a branch where it could be, it was the worst of times, or it was the age of, and then kind of goes on to the next part. And these really can be combined into this kind of branching web of frayed ropes and bubbles and spurs and branches that can start to get kind of complicated. Now, the goal of this, and I'll show you with nucleotides also, is to build this entire graph, simplify it as much as possible, and then perform a Euler walk. In other words, trying to cross each bridge only once in the best possible or most efficient route, and that will give you your sequence, your original sequence. So in this example, I've got arrows to kind of help you out. We can go through this and see that there's that kind of this bubble or this loop, and then it loops back down. There's really only one path through this where you do a Euler walk, where you don't backtrack. So I'm going to read it because each spot, each node is adding a word rather than a base pair. It was the best of times. It was the, we can go this way, worst of times. It was, loop back, the age of wisdom. It was the age of foolishness. And so we have reconstructed this repeat region where it was the, it was the, it was the. And actually, if you go back to the original paragraph, we've actually got it all right in the right order now. And so you could kind of continue this process. Same thing with nucleotides. You could look through here and go, it's ATG, ACC, TACG, ACGG, or AT, whatever. That's possible for a Euler walk as well. And that's how these assembly programs do it. Here it is a little more visually. We've got this unknown genome that we had these reads sequenced from. We broke it into k equals 4. And if you were to build a De Bruijn graph of this k equals 4 and perform a Euler walk, you would have to start here, loop your way around, and as you add all these bases, you'll actually reconstruct the genome's original sequence. So I don't expect you to memorize this. I just think it's kind of cool and helpful to understand what these things are doing. This is kind of an interesting way to think about assembling a genome, very different from alignment. And so again, just to kind of reiterate, one of the biggest problems here is that not all of this branching and converging and confusing map is actually error-driven. A lot of it is biological. There are repeats throughout the genome. There are inversions and all sorts of funny things that go on. And biology is complicated, and algorithms are going to do the best they can. But often this biological complexity is confounded by also sequence error and technical complexity. And so this really paints a very difficult picture for really large genomes. There are a number of software that try to deal with these as best they can. You can see them kind of listed here for genome assembly. They have some things that they tend to share, but they all do things slightly differently. Almost all of them try and reduce error by removing spurs. They try and collapse bubbles. If we're thinking about big genomes assemblies, we're not really worried about allele polymorphism quite right away, so those things get collapsed. And so when it's all said and done, what you end up with are contigs and or scaffolds. And so what are contigs and what are scaffolds? These are terms that get thrown around a lot, and I think it's important to understand. So contig is just shorthand for a contiguous sequence from reads. In other words, if you sequence a genome and you get all these reads or k-mers from those reads and put them together into a stretch of nucleotides that are longer than the reads that you started with, in other words, they're like a bunch of reads all kind of stuck together, that is a contiguous sequence from those reads, a contig. And so our goal is to get bigger and bigger contigs, eventually to the point where our contigs are pretty much the size of our chromosomes or genomes. Now, that is very difficult, again, because of repeat regions, because of complexity. And so often we don't have pure contigs for the entire genome. What we're able to do is scaffold contigs together into bigger chunks of good information about the structure. And so imagine these purple areas being large contigs. If we have these large contigs that are just kind of all disconnected from one another, But then we have sequence data that are paired in with large insert sizes, maybe a kilobase. And we have one pair mapped to one contig and another pair mapped to another contig, and we know the insert size and where they map to. Well, now we can start to bridge together these contigs, and we may not know the sequences in the middle in between them, but we do know the distance between them because of the insert size. And so we can fill those in with unknowns or ends. And if you look at any genome, even the human genome, one of the better eukaryotic genomes we know, it's full of ends. And it's full of ends because it's been scaffolded together to bridge repeat regions and other difficult things. But we know that there's something there, and we know how much of something is there, just not the exact base pairs. And so contigs and scaffolds are really what your assembly really is comprised of. If you're interested in assembling RNA for transcript related kind of analysis, it has its own set of assemblers because RNA transcription is not uniform across the genome. Some genes express more highly than others. And so you end up with all this kind of extra complication in that you have variance in coverage. Certain regions are going to get more reads mapping to them because they had more transcripts being produced. That is actually biological and not an error. So it requires a kind of a different way of thinking about assembly that we won't get into. Final thing I want to touch on are just some kind of cool assembly tricks to think about when you think about how library prep and read length and things all play into getting a good assembly. We end up with our De Bruijn graph that's very complicated. We do have some tricks to go back to get rid of our k-mers after we put our De Bruijn graph together and go back to our longer reads and use them to try and clean things up. If you've got this frayed rope situation, if you can find a read that maps to this frayed rope and bridges the distance of it, or you could then actually pull it apart because you go, well, if this read starts here and ends here, we know that this is its own, unique stretch. And so we can kind of disentangle frayed ropes. We could disentangle frayed ropes if they're too long to bridge with a read, but we've got paired end reads that hit either end of one side, we could disentangle it as well. Or if we know the distance between our paired reads, if we have a set insert size, and the two branches are of varying lengths, one length might be the only possibility between our two mate pairs. And so this is another way of collapsing bubbles. When this is all said and done, you end up with a bunch of contigs and scaffolds in a FASTA file. How do you know that it's good? And this is really one of the most difficult things to really wrap our heads around because you don't necessarily know everything because no one sat down and just read through a genome because we don't have that technology or the time to sit there and read 50 billion base pairs. So the actual correct answer is always unknown. We can do our best to try and assess how close we think we might be. But unfortunately, many of our assessments are actually based on the continuity and the size of our contigs rather than the content of them. We assume that that's going to be okay. And so often things that you'll see thrown out as metrics for how good an assembly are this number N50, the number of scaffolds, and the number of total bases contained within all your contigs and scaffolds. And so I'll walk through what those are. So the reason we talk about number of scaffolds is because the fewer scaffolds your genome's contained in, the bigger they must be, the better your assembly must be. In a perfect world, if you have 5 chromosomes and I sequenced and assembled your genome, if I did it perfectly, got everything right, I should have 5 scaffolds, right? Chromosomes aren't connected. So every scaffold should basically be representative of a single chromosome. In other words, the number of scaffolds, the goal is to make it the number of chromosomes. This very rarely happens, but it is possible. The next thing we want to do is look at the number of bases contained in your assembly. If you think of this assembly as being your entire genome, even if it's broken up into more scaffolds than there are chromosomes, the goal is that all of the pieces are actually represented, whether they're connected or not. There are many ways to estimate the genome size of organisms that is not a assembly metric. There are fluorescence methods of quantifying DNA, things like that. And so we have a good sense of the total bases that should be in a genome. And so we can compare. Here you can see a Staph aureus genome size is just shy of 3 megabases. And you can see the total number of bases assembled by a couple different assemblers. Here you can see Abyss2 actually did a pretty poor job. Abyss2 almost assembled a genome that was twice as big as it should be, whereas Soap de Novo seemed to do a pretty good job and hit right on the button. Same with all paths as well. So this is another way to kind of assess the quality of your genome assembly. Finally, relates to thinking about number of scaffolds, things like that. On average, how large are your contigs or scaffolds that your base pairs, your genome, are contained within? You don't want them all contained in a bunch of 20 base pair pieces. That's a lot of really tiny puzzle pieces, a lot of fragments. It'd be better if it was in 40 pieces, something like that are huge. And so people use this number N50. And what N50 is basically the size of the contig, the minimum size of a contig, that if you picked that size and up, that it would contain 50% of your assembled base pairs. And so here, if this contig in the human genome is 46 million base pairs long, and you counted all the base pairs in this contig and every contig larger, it would contain at least half of the assembled bases. That's what the N50 number is the size of that contig that's basically the median that contains the median base pair, the very middle point, if you line them up from smallest to largest contig. Doesn't mean that they're right, but the higher your N50, the larger your contigs on average are across the genome. So kind of big take home here, genomes are complicated. Things that help with assembly are having lots of depth, lots of reads to map. The more reads you have, the less sequencing error becomes a problem. Read length is important. The longer the reads, the more trivial certain repeats are. Insert size, the bigger your inserts, or even a variability in inserts, allows you to better bridge repeats, better scaffold contigs together. Kamer size varies with your organism. It's something you have to figure out on your own. And then again, sequencing accuracy along with read depth kind of play a role in how confident you are in the bases that you're assembling. So hopefully that was kind of shed some light on how genome assembly works, and you'll be able to practice it in lab.